For this video, we want to begin our discussion of cell respiration. And this is an essential process that allows organisms to be able to survive, as it provides energy that cells need to be able to carry out their functions. And we'll split this video into a number of different parts, and so this is C1.2 part 1. The type of energy that cells need to be able to carry out their functions is called ATP, or adenine triphosphate. And this is a molecule that's similar to DNA and RNA in that it's a nucleotide because it has a base of adenine, a 5-carbon sugar, ribose, and it has 3 phosphate groups that are the phosphate groups are in a chain and they're negatively charged. And it's oftentimes, this molecule is oftentimes described as the currency of cells because it is what allows the cell to be able to carry out its purposes, its functions. And it has a couple of specific properties that allow it to do this. Firstly, it's soluble in water, so it can move freely throughout the cytoplasm. It's stable at pH levels that are close to neutral. It cannot pass through the phospholipid bilayer, and so that helps to ensure controlled movements. The third phosphate in the chain of 3 can easily be removed and reattached by hydrolysis and condensation reactions, and the splitting off of that third phosphate through hydrolysis releases a small amount of energy. And this release of that third phosphate is what provides the energy for cells to be able to do work. The use of ATP falls under 3 kind of broad main categories. And first would be the synthesis of macromolecules. Anabolic reactions in which monomers are combined to form polymers requires ATP to be able to carry out that coupling, that combining. And so that would be the first use. The second use would be through active transport, the pumping of ions and particles across the membrane against the concentration gradient. It requires the pump to change shape to be able to facilitate that movement, and that changing of the pump requires energy, and that energy comes from ATP. The pump itself, when it changes its conformational shape, is one form or the other is more stable and the other is less stable, and so the use of ATP changes the shape of that pump to a less stable shape, and then it will revert back to its original without the need of energy. So one ATP can provide the change and then the revert back to the original. The third is movements of materials within cells, changing the shape of the cell, like during the cell division process, movement of cell from place to place. For example, white blood cells moving to a site of infection, muscle cells contracting and moving, etc. So synthesizing molecules, active transport, and movement are the 3 broad categories of uses of ATP. ATP as a molecule has 3 phosphates, and when it releases a phosphate group, it becomes ADP, which stands for adenine diphosphate. And ATP has more energy than ADP because that phosphate group hasn't lost. And in the conversion of ATP to ADP, energy is released. It's small, but it's a small amount of energy, but it's sufficient for powering or carrying out the reactions within the cell. When that energy is released, it's often used to be able to change a molecule, change the shape of a molecule. For example, we talked about the conformational shape of the pumps. The conversion of ATP to ADP is not 100% efficient. Some of that energy is lost, and it's lost in the form of heat. And in order to add a phosphate group, a process called phosphorylation takes place, in which phosphorylation is the adding of a phosphate group to ADP to be able to make it ATP. The opposite of that would occur through hydrolysis, in which the ATP is split apart, releasing energy. And the energy necessary to be able to produce ATP, the conversion of ADP to ATP, can come through a couple of different steps or mechanisms. Probably most commonly, and what we're going to spend a lot of time looking at, is cell respiration, the oxidation of carbohydrates, fats, or proteins to be able to form ATP. Additionally, some ATP is produced during the process of photosynthesis, although that's not its main objective. It primarily produces sugar. Light energy is used to convert into chemical energy. And then the third, and probably less common, or not one that you would necessarily think of, is chemosynthesis. And this is the oxidation of inorganic substances such as sulfides. And this would be a process that would take place, that we would see taking place in archaea. The quantity of ATP in a cell at any one point is often pretty small. And once it's used up, it requires whatever the action that the ATP is being used for, it stops that action. And so, for example, if neuron cells run out of ATP, they're unable to carry or convey impulses, and muscle cells stop contracting, causing or resulting in a cramp. Eventually, if the ATP is unavailable, if more is not being produced, the cell degrades pretty quickly, and then the damage is soon irreparable, and it leads to the cell death. And then if that were to occur continually, there would be a lot of problems for the organism. It would not be able to survive because ATP is essential for cells to be able to function. Cell respiration is the process in which carbon compounds are oxidized to release energy in the form of ATP. It's performed by living cells. Glucose and fatty acids are the main compounds that are used. For humans, we acquire these by consuming food. Plants produce glucose through the product of photosynthesis, so that's where they're getting their energy. And this process must be continually performed in order to produce ATP. Cell respiration differs from the process of gas exchange, and these two can be misconstrued or overlapped sometimes. Gas exchange is the intake and the release of gases. We're intaking oxygen and releasing carbon dioxide. Both of those are related to cell respiration, but the exchange of those gases is a separate process from cell respiration. Many cells use oxygen during respiration and produce carbon dioxide as a waste product, as we'll see as we progress through our explanation of respiration. These gases move across the plasma membrane independently, and they do so by simple diffusion. This process is not the same as respiration. Gas exchange is necessary for respiration and vice versa, but respiration creates concentration gradients that causes gases to diffuse. So the exchange of gases would not be able to be taking place because those concentration gradients wouldn't be developing or formed without the process of respiration. So they are linked, but they are separate processes, respiration and gas exchange. Cell respiration can be performed with oxygen aerobically, as well as anaerobically with no oxygen, humans included in this. Aerobic is the ideal. Oxygen is necessary, and it serves as an electron acceptor in the oxidation reactions. We'll talk about that momentarily. Carbohydrates, specifically glucose, lipids, fats and oils, and even amino acids sometimes can be used to help facilitate aerobic respiration. CO2 and water are the waste products, and the process results in a high yield of ATP, about 30 ATP per molecule of glucose. The initial reactions that kickstart this process occurs in the cytoplasm, but the majority of the reactions take place inside of the mitochondria. The opposite of that would be anaerobic respiration, so this is no oxygen. Other substances used as the oxygen acceptor. It only uses carbohydrates. In humans, it results in the production of carbon dioxide and lactate, and in yeast, it results in the production of ethanol, which is helpful for the production and synthesis of beer and wine. There's no water that's produced in anaerobic respiration. It has a low yield of ATP, only about 2 ATP molecules per molecule of glucose, so big difference compared to aerobic. And all of the reactions, there's far fewer, but they all take place in the cytoplasm. The mitochondria is not used for anaerobic respiration. If we know that anaerobic respiration eventually leads to cells degrading and dying, if that time period is long enough, why then does anaerobic respiration even exist for humans? Aerobic respiration, the use of oxygen, it generally provides sufficient oxygen to cells. But sometimes there are situations when oxygen cannot be delivered fast enough or efficiently enough, and in that situation, anaerobic respiration is beneficial. And this could be seen in like sprinters or other activities that would be really high intensity for short time periods. And so anaerobic respiration has some advantages. It can supply ATP very rapidly over a short time period when the maximum power of muscle cells and muscle contractions is needed. From an evolutionary standpoint, you would think like escaping from a predator or trying to catch a prey. Short time periods of intense sprinting, and probably in situations where oxygen intake can't keep up with the demand for aerobic respiration, and so anaerobic respiration would then be used. Other examples would be like intense weightlifting, short distance runners today, sometimes long distance runners, cyclists, and rowers during the sprint finish of their race. And so it is beneficial to have this ability to produce ATP when oxygen supply is limited. When this does take place, lactate or lactic acid is a waste product, and it limits the limits to the concentration of the amount that the human body can tolerate, and it limits the amount of anaerobic respiration. So the buildup of this actually then limits the amount of anaerobic respiration that can take place. It must be broken down after it's produced, and in order to break it down, it requires oxygen, and it takes time, several minutes. And so this is called an oxygen debt. So the process of anaerobic respiration would need to be a short time period, because one, cells won't be able to survive without oxygen, and two, the production of lactic acid eventually needs to be broken down, and that requires more oxygen. It's quite possible for a rate of cell respiration to be able to be measured, as well as looking at different conditions and how they affect the rate of cell respiration. We'll actually do this in my class. If you're in a different class, I would recommend that you investigate some other methods or try to do this within your class. The rate of cell respiration can be measured in a couple of different ways. One would be oxygen uptake, so we're talking about aerobic respiration in this situation, the production of carbon dioxide, or the consumption of glucose or some other substance. The measurement requires, to be able to measure this rate, requires a specific apparatus called a respirometer, and this is a sealed glass or plastic container in which the organism is placed inside with sufficient air to be able to remain healthy. We don't want that organism to be harmed, and a base is used to absorb excess carbon dioxide during the process of respiration. So that organism would be producing carbon dioxide, something needs to be able to absorb it. And then a capillary tube containing fluid is used to be able to measure the change in volume of air inside that respirometer. So then we can measure the rate of how quickly this is taking place. The measurement should be in units of volume, and to determine the rate, it must be in relation to time. So an amount of volume change per time. So it's possible then to investigate a number of different variables and see how they affect the rate of respiration. For example, comparison of respiration rate of different organisms or different types of organisms, the effect of temperature on respiration rates. Aerobic respiration involves numerous enzymes that help to facilitate the process, and as we know, temperature and pH can impact those enzymes. The third potential would be a comparison of respiration rate of active versus inactive organisms. And so those would be some possibilities of things that you could investigate to see how different conditions affect cell respiration rate. Another possibility for measuring the rate of respiration is the use of Vernier gas sensor probes. Vernier is a company that makes a number of different scientific measuring probes. For example, just simple gas pressure as well as oxygen and carbon dioxide. And they also have some sealed chambers called biochambers that can be used to be able to put inside some organisms and then measure the gas changes during the process of respiration. So those can also be used and help to facilitate some good IA options. As we begin to get into how this process of cell respiration actually takes place, we need to understand the role of a molecule called NAD. And in order to understand this, we need to have an understanding of the process of oxidation and reduction. And these two processes occur concurrently within a reaction as reactions are taking place, so they're happening at the same time. Oxidation is the loss of electrons from a substance, and this can occur through the loss of a hydrogen atom from a molecule or the gaining of oxygen, because oxygen has a high affinity for electrons. It pulls electrons towards it. Reduction is essentially the opposite. It's the gain of electrons, and this can happen through the accepting of a hydrogen atom or the loss of an oxygen atom. Electron carriers are types of molecules or substances that can accept and lose electrons reversibly, so they can add or subtract. And so electron carriers as molecules link oxidation and reduction reactions together. NAD is one of the primary electron carriers in the process of respiration. It is a molecule that, when it acquires 2 electrons, becomes reduced NAD. The molecule NAD is initially positive, and it accepts 2 electrons and 1 proton. That proton would be from a hydrogen atom from an oxidized substance, so a substance that's being oxidized, and that then creates the molecule NADH, as well as a hydrogen ion that gets released. And this molecule of NADH is essential to carrying out cell respiration, as we'll see a little bit later on in our steps, as well as those hydrogen ions are also essential to be able to facilitate this process. And so NAD is necessary. It is an electron carrier that's necessary in the process of aerobic cell respiration. The process of cell respiration begins with the process of glycolysis, and this process takes place in the cytoplasm and serves as the first step, the first metabolic pathway in this overall metabolic pathway that produces ATP. And so for aerobic respiration, this is also the first step, same for anaerobic. It usually uses glucose. Takes place in the cytoplasm, and the conversion of glucose to pyruvate, that's the final product, occurs through the use of multiple enzymes. So this is a great example of a metabolic pathway, a biochemical pathway as we've discussed. It produces a small yield of ATP without the use of any oxygen, but that's not its primary purpose. Its primary purpose is to produce this molecule called pyruvate. And the process can be broken up into 4 different stages. The first stage is a phosphorylation of glucose. And phosphates, or the process of phosphorylation, is the addition of a phosphate group to a molecule. It requires a little bit of energy, but it improves the stability. And usually that phosphate comes from ATP, though not always, as we'll see here in the case. But for this initial step, a phosphate is linked to the 6 carbon of glucose through phosphorylation, so ATP is used, and that glucose is then converted to fructose. It helps to make the molecule more stable. It's also symmetrical, so that the molecule can be split. A second phosphorylation takes place, ultimately producing a molecule called fructose 1,6-biphosphate. It's a fructose molecule, and it has 2 phosphate groups on it, carbons number 1 and carbon number 6. The second stage is a Lys stage, or a breaking stage, and this is when that fructose biphosphate is split into 2 different molecules, and it forms triose phosphate. And so each of those molecules have 3 carbons in them. It's kind of helpful to keep track of the number of carbons. Fructose has 6 carbons, and so it gets broken into 2, and each of those triose phosphate molecules have 3 carbons. The third stage is the oxidation stage, and each of those triose phosphates oxidizes or becomes oxidized by the removal of hydrogen, loses the electrons as well, and they bind to NAD, which becomes reduced, forming NADH, as well as some hydrogen ions. The energy released by the oxidation of the triose molecules allows a second phosphate group to become attached, and that phosphate group is actually not coming from ATP, so it's not using more ATP. This produces a molecule called 1,3-biphosphoglycerate. It has 2 phosphates on it, carbons number 1 and carbon number 3. The fourth stage is the ATP formation, and this is the transfer of phosphate groups to ADP to produce ATP. It occurs twice, so we've got 2 biphosphoglycerate molecules, and each of those biphosphoglycerate molecules has 2 phosphates, so it occurs twice per molecule. The result of this then is that organic molecule, that 3-carbon triose phosphate, or biphosphoglycerate now, becomes pyruvate, and those 2 pyruvates are produced from 1 molecule of glucose, because those pyruvates still have 3 carbons each. So the overall outcomes of the process: the 1 glucose molecule is converted into 2 separate pyruvate molecules that each have 3 carbons. 2 NAD are converted to reduced NAD, or you could call them NADH. 4 ATP are produced, but we used 2 at the beginning before that molecule was split, so our net yield of ATP is 2 ATP molecules, and no oxygen is required. Those pyruvate molecules are then going to go on into our next step called the link reaction that we'll talk about in our next video. The NADH is going to be used in a later step called the electron transport chain, and the ATP is produced to be able to help the cell carry out its functions. And so in our second video, we will take a look at the remaining steps of this process of ATP formation and cell respiration.